Welcome back to part 2 of Nutritional Adaptations lecture. The second microbial association or symbiosis that we're going to look at is that of mycorrhizal fungi associated with plants. Now, this is really common. Many trees and plants have mycorrhizal associations. They're actually quite considered very ancient symbiosis, and that virtually all plants have these associations, over 90%. So we covered some of this in the fungi lecture, but let's go through the major ideas again. As I said, very common. This picture here shows you the a seedling here, and you can see it's a little pine tree, and this is still photosynthetic, so the plant is doing photosynthesis, and this region here is where the roots are, and then beyond the region at which the roots have kind of exploited, you have this huge network of mycorrhizal hyphae. That's the hyphae of the fungus that is associated with the plant. So in order to set this up, there is a very specific association between a plant and a fungal species. So not just any mycorrhizal fungi associated with any plant, they have very specific genetic interactions. And you can see the fungus forming this web as the seedling grows. The mycorrhizal gets a bigger and bigger volume of soil, which is exploited, and nutrients all the way out here can make it to the plant roots, whereas the plant root volume is volume of soil that the plant's roots are actually connected with is much smaller, and so this helps take up and exploit nutrients in a much larger volume of soil around each plant root area. So you might remember that the fungi are really good at taking up nutrients, especially things like phosphorus and other minerals, and they take up this inorganic nutrients and then sort of essentially shunt it through the hyphae to the plant roots. So in exchange for this, the fungi receives organic carbon or sugars that have been made in the green part of the plant, in the leaves, through photosynthesis, and then transported down through the stem via the phloem, and then out to all of the little roots, and then there's an exchange, space for exchange inside the root tissue, where the plant gives up that organic carbon and receives the phosphorus and other minerals in exchange. So organic carbon is traveling downwards, phosphorus, nitrogen, and other minerals is traveling from the fungus into the plant, and then from there up to the rest of the above ground part of the plant. So much like the this diagram is much like the one that we looked at for legumes, except this is showing how it's established, how that relationship, that connections is set up between the fungus and the plant roots. So the plant tissue was shown here, cortical cells here, the epidermal cells shown in yellow, and somewhere in the soil there might be a spore or fragments of hyphae, and then as the plant seedling starts to grow, they can come in contact with each other. This can involve a whole lot of chemical signaling, and then you can see the fungus actually growing in between the root cells here and establishing little connections inside the root cells, which have a high surface area for exchange of those nutrients. And then that network is connected all the way for some volume out here into the rest of the soil to take up nutrients and pass them to the plant. So this diagram kind of shows you a little bit of that. You remember that fungi do external digestion, so fungal hyphae release enzymes. This works on organic materials and breaks them down, releasing things like nitrate and phosphate, and then that gets taken up by the fungal hyphae into the hyphal cells and then transported through the hyphae all the way to the hyphal connections inside the plant, We have a couple of different types of hyphal connections shown here. In this lower one, you see a really high surface area of the red fungus releasing nitrate and phosphate that it's taken up some distance away from the plant up here, and then taking up photosynthetic products that have come down from the plant into the roots and then are passed to the fungus. And then the fungus takes those and then transports them out to the rest of the fungal cells to support that mycorrhizal growth. So the fungus is taking up nutrients that the plant can use. Just a reminder, fungi do not fix nitrogen. They do not fix nitrogen gas. This is only done by bacteria, as we saw in the earlier section of this lecture. So that seemed to be a misinterpretation that students often come up with, and I'm not sure where it comes from, but that's just a reminder there. So plants, so plants are providing the fungus with fixed organic carbon. The other thing that this association can do is that it can protect against pathogenic fungi or bacteria that might be hanging around in the soil. Just having that fungal network around them is going to protect the plant roots from attack of those pathogenic organisms. So the advantage to the plant, it gains nitrogen and phosphorus and micronutrients from the soil that it can't reach with its own roots. The advantage to the fungus is, remember, fungus is a heterotroph. It needs to get organic carbon from somewhere else, and so it gets sugars that have been made by the plant as its food source. This diagram here shows you, again, that kind of exchange and how we kind of can analyze or understand how this process goes on using experiments. And one of the ways to do that is to use a tracer, a radioactive isotope tracer. So we have a radioactive CO2 that's a carbon 14 label on the CO2. And this is fed to the leaves. That CO2 is fixed in the leaves by photosynthesis into organic carbon, sugars. And so now we have that radioactive label in sugar. So we've got to have radioactively labeled sucrose, glucose, and other sugars inside the plant. And at the same time, we're also providing radioactively labeled phosphorus and nitrogen to the plant, to the fungus, not to the roots, but just to the fungus, which is some distance away from the roots. The fungus takes up that radioactively labeled phosphorus and nitrogen and then shunts it through its hyphae and eventually passes it on to the plant through those connections between the hyphae and the plant cells. So in the intermediate, we have radioactively labeled carbon compounds in the plant tissue. We have radioactively labeled phosphorus and nitrogen compounds in the fungal tissue, shown here in green. And then over a period of time, as these two partners exchange nutrients with each other, we get the radioactively labeled phosphorus and nitrogen appearing in the leaves of the plant as it's being transferred through the root tissue up through the xylem and to the leaves. And at the same time, the fungus is also gaining that radioactively labeled carbon that's coming from the plant leaf tissue that comes down through the roots and then is passed to the fungal partners. Okay, so we're going to move on also. The third one is an organism that is parasitic. Now, you're probably familiar with parasites as terms of pathogens. Parasitic plants are really cool because they do a lot of things that normal plants do, in that they, for example, they're photosynthetic. One of the examples is mistletoe. Now, mistletoe, of course, has that whole idea, you know, kissing under the mistletoe is supposed to be very romantic, and yet this is actually a parasitic plant. So I'm not quite sure what that means about human relationships, but I'll leave that up to you. So these plants are photosynthetic, but a seed lands on the stem or the trunk of another plant, and it grows down, and then it grows its tissue into the host plant, and then connects into the xylem tissue of the host plant in order to access nutrients and water that are coming through the xylem of the host. So here on the left you can actually see a transmission, actually a light microscopy view of the juniper. Here is the host plant, and you can see the clear tracheids and a few vessel elements in the stained blue. And then this tissue here is actually from the mistletoe, and this is pushing down through the juniper host bark into the vascular tissue of that host, and from there it gets access to xylem flow and the nutrients and the water that are in the xylem. So there's another few parasites, plant parasites that are kind of well known. One of them is called dodder. So here we have a host plant here, stem, and then this tendril essentially is the dodder plant, and it winds its way around the host tissue like this, and then between the host tissue and the parasite, you can see here there are some host connections. So those other kind of tissue places where the parasite sort of essentially pushes tissue down into the host to connect into the vascular tissue and get access to nutrients. This is ultimately not a particularly good relationship for the host plant. You can see in this picture here, this dodder has almost taken over the tree. It's sort of hanging off it, almost like a kind of hair. And ultimately, over many, many years of growing like that, it will probably kill the plant. So this is not a mutualism. This is a one-way street. The parasite is getting the benefit here. Carnivorous plants are the last group I'm going to talk about. This is close to home for me. I work on pitcher plants. And carnivorous plants have evolved several times over evolutionary time. And they include a range of different mechanisms, including pitcher plants. This picture down here, this is a Nepenthes, a tropical pitcher plant, which produces pitchers on the end of its leaves. We also have a classic Venus flytrap that we talk about in the lab a little bit in terms of the movement. And then we also have a Drosera here, a sundew. And you can see it's managed to catch a fairly large insect, which it will slowly digest. So just a point, reminder that carnivorous plants are also photosynthetic. You can see the green tissues on particularly this Venus flytrap. It is still able to do its own photosynthesis, so it's still making organic carbon from CO2. But it captures insects and digests them to supplement its nutrients needs that it's not getting from the soil, so nitrogen, phosphorus, potassium. And this is because it grows in nutrient-poor habitats, things like wetlands or boggy areas, tropical rainforests that are poor in nutrients, and often sand plains too. Remember, sandy soil is not very fertile. So these are all examples of carnivorous plants that have adapted to getting nutrients in a different way. So this is carnivorous plant Drosera. You can see a whole range of different structures that it can produce to capture insects and then digest them. And here's a Drosera that's been rather ambitious and managed to catch a dragonfly. And up close, you can see the hairs here that have a blob of digestive enzymes on the end. This is what they look like up close, the hairs, and then the digestive enzyme secreting structures right on the end there. And then over a period of many days, maybe even weeks if you have caught something this big, the plant will slowly degrade the insect and then withdraw the nutrients, take them up into the plant tissue. Just a reminder also that carnivorous plants are all flowering plants, so they also need to attract pollinators. So here's a range of different carnivorous plants while they're flowering, and you can see they've not only got really attractive flowers, but these are insect pollinated flowers. So they're going to be attracting an insect somewhere on the for pollination on the flower, at the same time they might be capturing other insects to digest for as to supplement their nutrient needs. You notice also that many of them have fairly long stalks for the flowers. So this is helping to separate the flowers that need pollinating with their sources of insects that might be caught in the traps. Okay, so just a little wake-up question here. Which of the following nutrients are likely to be gained from carnivory in plants? So this goes back to that active learning exercise that I talked about. It's like, where are different nutrients coming from? Which ones are macronutrients and therefore going to be needed in large concentrations? Which ones are micronutrients and therefore probably less likely to be limiting in soils? So let's have a look at this, carbon and oxygen. So where do plants get carbon from? You remember all carnivorous plants are photosynthetic, so they're going to be getting carbon from carbon dioxide that they fix through photosynthesis. And oxygen is either going to be coming from carbon dioxide or from water. Molybdenum and copper, these are both micronutrients needed in small concentrations. So the chances of these being limiting in soils is fairly slim. Iron, magnesium, and carbon. Okay, so we unpack this. We've got a few here. We've got iron is a micronutrient, so less likely to be limiting. Magnesium is a macronutrient, so that could be limiting. And then carbon they're getting from CO2. So not all three of these make sense. So that's not a good option. Nitrogen and phosphorus. Are these macronutrients? Yes. Are they nutrients that are normally gained from the soil? Yes. Therefore, that makes sense that they would be gained from carnivory in plants. And then this last one, water and carbon dioxide. Carbon dioxide, of course, is coming out of the atmosphere and fixed through photosynthesis, and water is just being taken up from the soil. That's not really a limiting factor. If it were that limiting, the plants probably wouldn't be surviving anyway. Okay. So there's some little prompts there for things to think a little bit about in dissecting this question. So I wanted to finish this with a link to a video. This is a fantastic video. It comes from David Attenborough's BBC series, The Secret Life of Plants, and it takes you through the functions with some great photography of a few different types of carnivorous plants. So I normally would play this in class, but you can link to that yourselves and have a look. I highly recommend it. To summarize, plants are often nutrient limited in most many ecosystems. In agriculture, we have to add fertilizer, but we're probably going to run out of phosphorus to make critical fertilizers in order to improve food production. And this is going to be a crisis as our world population increases and we keep needing to feed more people. But plants have been dealing with this problem for a long time. They have a range of nutritional adaptations, including nitrogen-fixing symbioses that we see in legumes, mycorrhizal associations with fungi, parasitism, and carnivorous plants. And then I just wanted also to remind you that I work on these carnivorous plants, these pitcher plants, here growing at the UWM field station, which is about 45 minutes north of our campus. It's a fantastic resource. There's a large, almost pristine wetland there, which has a whole range of wetland plants. And we work on, myself and my lab works on the nutritional ecology of this pitcher plant, Sarracenia purpurea. There's often summer research projects. The opportunities for undergraduate research are a little limited at the moment through COVID, but we're hoping that these are going to open up again fairly soon, maybe in spring. So if this sounds interesting to you, let me know and I might be able to incorporate you in some of our lab research. Okay, take care and I'll see you in the next lecture.